everybody, and welcome back to Fossil Friday Chats. I'm Brittany Stoneberg from the Western Science Center. With me, as always, is my co-host, Gabriel Santos from the Alpha Museum.
TGI Fossil Friday, everybody.
And joining us today is someone I'm very excited to meet and talk to about their work, Dr. Ingrid Romero. Hi, how are you doing?
Hi, good. Thank you for the invitation.
Oh, we're
Yeah,
so
we are.
excited to have you today.
Yeah, we are very excited because when you submitted information about your talk, you mentioned that, when we think about paleontology, we always think about macro paleontology. We think about big things. But today, we're going very, very small into the world of micropaleontology and palynology, which
Yes.
I know nothing about. So, which means I'm
Me neither.
excited to learn more
I know
about it.
zero
Yeah.
about it. Oh, no, I know it has to do with pollen, I think, but other than that, I'm very excited to learn about this stuff.
Yeah, no. And I'm happy to be presenting what is happening in the microscopic world of the fossils.
Yeah. If you're not familiar with Dr. Romero herself, she is a Colombian biologist and palynologist interested in understanding past environmental changes. She did her PhD at University of Illinois and throughout her career has been working with microfossils from the Neotropics, especially the last 65 million years. Her research includes a study about the marine incursions in the Cenozoic of northern South America and the application of machine learning on fossil pollen identification. That's very cool. I didn't know you could do that. She is currently a postdoctoral associate working on a project about fungi in a warmer world at Morehead State University. Yeah, this sounds really cool. Like you said, we always kind of think of paleontology as dinosaurs or big charismatic ice age megafauna. So we're going into very teeny tiny stuff today.
Well, not just that, but just a quick preview, but usually we think about paleontology, we're just like the Indiana Jones type in the field and the rocks. But you're going to talk about machine learning. This is like the future. This is like Mass Effect level types of science, and I am excited for it.
Oh. All right. Well, if anybody has questions for Dr. Romero, as always, go ahead and put them in the chat. We will have a Q&A session after her presentation. So, Dr. Romero, whenever you're ready, go ahead and tell us all about the micro world of paleontology and palynology.
Well, thank you so much. Let me share the screen. It's one. Okay, everybody is seeing it?
Yep.
Perfect. So, yes, hi everyone again. And again, it's a pleasure to be talking here in Fossil Fridays about microfossils. But a little about me before the start. So although Brittany already gave an amazing introduction, is that yes, I'm a Colombian biologist and palynologist, and I just finished my PhD in plant biology. And here I just want to put some photos of what someone in palynology does to do research. So one of those things is we have to go to collect samples of rocks or from the air. And but we also have to do some lab work, and most of the time it's going to be a lot of microscope work. So here there are some photos from Colombia, from Panama, and from United States. And here I also want to show that for any project in science, there is not just one person, it's always a team of people working together, and that's how we get the cool discoveries in science. So my passion about palynology started the first time that I saw a pollen grain in the microscope. And what it made me fall enchanted was the symmetry, the morphological variation that we can get, and also the diversity in size, in shape, in ornamentation, like arrangements in the wall. So that's how all my passion through palynology started in my undergrad, and it has continued till now. And I'm going to explain better what palynology means later. So now, why the title? Palynology, the hidden world of paleontology. Because when we hear paleontology, as Brittany mentioned, we think about macrofossils, right? Dinosaurs, big leaves, big bones. But we forget that always in the rock, we also have tiny, tiny fossils that will be the microfossils. And here we have a huge variety of remains that came from plants, like pollen and spores, from fungi, from aquatic organisms like dinoflagellates and algae. And here is when palynology came to place. So palynology initially was described as the study of pollen and spores and dinosaurs, but that definition has been expanded through time to study also non-pollen fossils, such as fungi, other type of algae. And the importance of palynology, or the study of the microfossils, is that the microfossil record is more continuous in a temporal and a spatial scale than macrofossils. So we can get a most continuous record, but also it can help us to understand or to learn more about past environments. So for example, past vegetation, past depositional environment. So how it was a lake, it was a marine environment, it was a fluvial environment. And sometimes some microfossils can give us information about the age of the rock. How old is this rock? Depending of the time that they appear in the record. In this way, so with palynology, we can answer questions like how were the environments in the past, how they changed through time. So for example, there was before a forest, then a savanna, then I don't know, there is a mountain and a more like a paramus, like high mountain vegetation. Which factors play a role in these changes? Like climatic, like temperature and precipitation, or geological, like for example, an uplift event. And how these past changes can give us information to make better predictions or make better inferences about how future climate changes can affect the ecosystems. So that's how here I'm going to show 3 cases of study. So my focus of study has been the Cenozoic, that is the last 60 million years, as we can see here. And I will be talking about marine incursions, a case of study with a specific fossil pollen type, and about fungi. So in the first case of study, the main characters are dinoflagellates. So here we have that dinoflagellates are the ones that produce that bioluminescence effect in the shore when we are in the sea. And they are mainly plankton, like algae, and they are usually indicators in the fossil record of marine conditions, although some of them could be associated with freshwater environments. And the dinocytes is just the stage of the dinoflagellates that gets fossilized because it's a dormant stage. So they were important for this study. So we did a study led by Dr. Carlos Jaramillo from Smithsonian about marine incursions in North and South America during the Miocene, between 23 to 5 million years ago. So first of all, what is happening in South America? So nowadays, South America has the neotropical rainforest that is considered one of the most diverse habitats in the world. And we have the Amazon basin, right, that we all have here for the high diversity. However, in the Miocene, we have information that it was flood, that all that area was flood, and it was a mixture between marine environment and freshwater, like lacustrine environment, but we don't know what was the timing of those flooding events. And that is important to understand why this place, why the Amazon is as diverse as it is today, right? So the question that we wanted to answer is, what was the timing of the marine incursions in North and South America during the Miocene? And for that, we study two geological sections, one from the eastern of Colombia, that is in the Llanos Basin, and the other in the Solimões Basin, that's the name of the Amazon Basin, that was more located in Brazil, limiting with Colombia, around here. So from that, we studied the microfossil composition and the geology, and we found a high representation of dinoflagellates and some of freshwater algae. But there were two groups, like they were this massive event where dinoflagellates representation increased, and it was here and here, which was in the early Miocene and in the middle Miocene. And here we can see some of the dinoflagellates that we saw. So basically, this was indicating us two major flooding events in North and South America. The first one in the early Miocene was shorter than the second one in the middle Miocene. And with the composition that we had, and especially with the dinoflagellates, this suggests us that the environment was more like shallow marine waters, and that the events were longer in the Llanos Basin than in the Amazon. So those were the two main conclusions of this study, where dinoflagellates give us kind of the answer. For the second case study, the main character is pollen. And what is pollen? Pollen basically is the sperm of the seed plants. But the cool thing about pollen is that each group has a unique morphology. So each group of plants have a unique morphology in pollen, right? Will be in size, in shape, and ornamentation. And that is important because that's what is going to give reproductive strategies for plants. But for us, in the fossil record, it allows us not only to reconstruct the vegetation that was in the past, but also to understand better the evolution of the different plant lineages. And for this second application, here, I will be talking about the reconstruction of the history of this fossil species that is known as Stratopolis catatumbus. And the main question that we wanted to answer in this study was, what does pollen tell us about the evolution of plant lineages when we improve the taxonomic identification? So usually pollen, we identify it to family or genus, mainly to family, but if we want to constrain that identification, we can have better ecological interpretations. And that's important to know about fossils, that to know what happened, we try always to find a modern analog. So here we apply machine learning models. So, and specifically convolutional neural networks. What this means? So basically, these models have a principle similar to the software that do face identification. So basically, they are very good processing visual data and extract morphological characteristics that they're going to codify to get a final identification. So you put an image and at the end tell you this could be A, B, or C, and what is the probability to be each one of those. So here in the example, we have that we put a photo. We're going to imagine we don't know it's a flower. And the models start running and going to select part of the photo, pixels or characters. And after hierarchical classifications, they ended in give us the potential candidates and the probability. So here we have that the higher probability is that the photo is a photo of a flower. So we did the same with the fossil. So first, who is Stratopolis catatumbus? This fossil has been found in the fossil record since the Paleocene, about 60 million years ago. Has a pantropical distribution, as we can see here in the animation, and has been associated to lowland forests. And it has been associated to a modern legume tribe that is known as Amherstia, that the modern members of this tribe are distributed in the pantropics, in the tropics, in tropical lowland forests. So here we can see some pollen grains of different genera of this tribe. And for our analysis, we select pollen grains of these 16 genera, and we use it to train the model, because these machine learning models needs to be first trained. Then you use a set of images that you have not used before to identify, to test the performance, and then to identify. So after we train the models with the modern specimens, modern pollen grains, we put our fossils. And for our fossils, we had 48 fossil pollen grains of this Stratopolis catatumbus from South America and Africa, and from different ages. So some were older and some were younger. And these were the results, but this is too confusing. So this is like a summary of the results, where we had that the African fossils that are the ones, the red point is the locality where the fossils were extracted. The African fossils were identified as modern genera that have a distribution that is pantropical or African. And the South American fossils were identified as genera that nowadays are distributed in around the pantropics or the tropics, or only in Africa or only in South America. And why this result was important? First, when we compare those results with the modern phylogeny of the tribe, which this figure is that phylogeny, what is important here is that we have the fossils of Africa and some of the South American fossils identified as modern African genera that are indicated in blue, or pantropical genera that are identified in white. And we can see that the African genera and the pantropical genera are older than the modern neotropical genus that is here indicated. And that's why only the South American, that is why, but it was also a good surprise that only South American fossils were identified as the South American genus. In addition, we have that there is also a consistency. This morphological variation not only have a consistency with the phylogeny, but also with the biogeography of the fossil. So here we have that the fossil first originate in Africa, and we're going to see that in a little, then later radiate through the tropics. So here you see first in Africa, then it start radiating in the tropics. So with that, we had that the pollen morphology was can give us more information about the evolutionary history of the plants that we previously expected. But the cool thing, that's something that cannot give us the phylogeny or the molecular studies, is that we can also see extinct morphologies. And I didn't show much about that, but some of the fossils have characteristics that were similar not only to one genus, but more 2 or 3. So like chimeras in the microfossil world, which is important. Now, in the third case of a study, I will be talking about fungi. Or that's the main character. So fungi are the main decomposers of the organic matter, and they are important because they help us to understand the carbon cycle, because some of them can retain a carbon cycle, but others can expel carbon. Carbon cycle, not carbon. The spores are some of the parts of the fungi that are preserved in the fossil record. So I started working in this project, Fungi in a Warmer World, in January, with as the head of the project, Dr. Jen O'Keefe. And the main question of this project is to understand how fungal assemblages change in response to global warming. So, and this is important to understand how they will be affected due to the near future climate change. So for this, we used to study in the past the middle Miocene climate optimum. So what the middle Miocene climate optimum occurred in between 15 and 17 million years ago, and it's a warming period of time that has been considered a potential analog of the future climate changes. And why is this? Because the mean annual temperature estimated for this time was about 13 degrees Celsius warmer than now, and because the atmospheric carbon concentrations are in the ranges estimated for the following 100 years. Also because the ice layers in the polar regions were reduced just because at that time they were forming. So that's why make it a good potential analog for the future climate changes. So for this project, the idea is to study fossils of fungi all around the world. Currently, I'm working with some samples from Thailand and from Slovakia, and soon I will start with some samples from Colombia and United States, but other colleagues are working with samples from Argentina and from China. So what part fungi can tell us about the environment and how we can relate this with past environments? So here I'm going to show you some examples. First, we have this fossil that we found in Philippines that has been associated. The name is germlings, and it's an immature stage of some fungi groups that has been observed in these different species of these two groups. But the important thing is they have been associated to humid environments or environments with high precipitation. We have also this cool spiral spore that has been associated to helicoma, and the modern helicoma has been found in leaves, wood, and has been associated sometimes with aquatic environments. We have this one that is one of my favorites that we found in Thailand and has been identified as Arecophila. And the cool thing of this one is that it has a very restricted biogeography, only found in Asia, in bamboo and palms. So as we can see, fungi can give us also information of which type of vegetation was around. Another one that I found in Colombia is this one called the Poroteca, and it has been potential indicator of lacustrine environments and has been found in some ferns and solanaceae plants. And here we have this one found in Australia, identified as Helicodermalista, and the characteristic is the type of sleep, this aperture. And it's been found mainly in New Zealand and Australia, and in wood, the wood of these two families of plants, Proteaceae and Lauraceae. And the last one, for example, is Tetraploa, found in Thailand, that has been associated to humid environments, but it's been always also found in dead remains of palms, grasses, and bamboo. So the idea with the project is when we have the fungal composition of the different places, we can do a paleoecological analysis and try to do a climatic reconstruction. That will be a diagram like this one, that it will tell us it was arid, it was humid, it was more tropical, it was more temperate. And in that way, then we can make model reconstructions. So with this, as final remarks, I just want to say that microfossils are cool. The microfossil record is more continuous in a temporal and spatial scale than macrofossils. They are good to have a better understanding of past environments, type of vegetation, the positional environment, and evolution of different lineages of organisms. They also help us to understand how these changes occur due to climatic or geological changes. And they also make allow us to make better inferences about how future climate changes can affect different ecosystems. And now, when you think about paleontology, I hope you not only think about macrofossils, but also microfossils. And last but not least, I just want to share with this or recommend you this book. It's in Spanish. It's about the paleontology of Colombia. The name in English will be A Long Time Ago, an Illustrated Trip of the Travel of the Colombian Paleontology. But this book, you can download it free in this website. And it also has cool illustrations showing fossils that have been found in Colombia and what we can say about the paleontology or the ancient environments in my country. So thank you so much for tuning in. Special thanks to the Museum of Paleontology and the Western Science Center for this invitation, and a special thanks to Brittany and Gabe for the invitation and for making this possible.
Oh man.
There you go.
Did I stop sharing?
You're good.
Yeah, you're good.
Perfect.
That was fantastic. I don't know why I forget sometimes, but like the fact that there's these cool fungi fossils out there that you can reconstruct what kind of plants there are, that really blew my mind today, because like I knew there were fungus fossils. I know fungi exist, but just that you can use them in such a cool way. I'm just like, my mind is blown right now.
Yeah, no, palynology is not only pollen and spores, include other groups. And fungi are a black box that many people want to open and understand better. Yeah.
When
So
you were getting
there are any
started,
questions?
well, I have a quick question.
Yeah.
So like when you were getting started in like, getting at your science career, did you expect yourself to be working in like these kinds of fossils and micropaleontology and fungi, plant biology, and combining all of this to kind of reconstruct the ancient climates and stuff? Is that what you set to do?
No, so I started looking pollen because I was interested to learn about plant-animal interaction, to work with pollinators, but from there I had the opportunity to do like in Colombia a pollen collection in my university. So I had to get class in other university and the training, and that's when I started working in more paleo. So I started learning about Holocene and how pollen can tell us all these changes. And then later I had the opportunity to work with Carlos in the Smithsonian, and that's when I went deep time. So yeah, but no, initially I wanted to integrate plants and animals.
That's cool. Here's a cool question from one of our viewers. This is from Alexa, who's 9 years old, asking, what is the smallest microfossil that you've ever found?
Oh, wow. The smallest microfossil is, there are some fungal spores that are less than 5 micrometers. That's very, very, very tiny.
Oh
that
my
you barely
gosh.
can see in a magnification of 100x. So, and you barely can see anything about them. But yeah, that was the smallest one.
That's crazy.
Really, it really is the microscopic world. I mean, for a lot of folks who think about paleontology, they think, like I said earlier, they think about us going out, finding these bones and rocks and digging them out, bringing them to the museum. But for you, what is the process like of finding microscopic fossils from the field to your microscope? Like, how does that go? How do you know where to look
So,
even?
so well, when you find the macrofossils, if you took a piece of that rock, will be like a tiny piece, and you put it in acid. Usually, like now there are methods that you don't use acid, but the traditionals we use acids that dissolve the rock and liberate the organic parts. And in those organic particles, we're going to find these microfossils. But there are also, so depending of the type of microfossil you want to study, you're going to use different chemicals and different processes. But if you took any piece of rock or any piece of soil and you put it in acid, well, the kids, please don't do this, you're going to find, but now if you take a piece of dirt, yeah, a little of dirt, and you put water and look at it in the microscope, you're probably going to find pollen that has fallen from the trees. So yeah, just or from the same macrofossil, you just scratch a little bit, and you're going to find something there.
That's cool.
Well, I'm going to be going to my museum collection and I'm going to start scraping all the fossils and seeing what I can find now and using our microscope.
Here's a question that always gets asked every week, and I'll be the one to ask it this time. What's your favorite fossil, like microfossil, that you've ever looked at or studied? Do you have a favorite child?
Yes, yes. My favorite shell, it was at the beginning of the presentation, is the pollen that is spiky, that is associated to a tropical family of plants. But overall, from my favorite microfossils are the spiky pollen. I just like them. And many of the families like Asteraceae that produce these spiky pollens, Asteraceae also the family of the sunflowers, many of them produce allergies. So they're not really nice, but they're beautiful and they're very complex. So yeah, that will be my favorite.
Yeah, that's my usual relationship with pollen is allergies.
Yeah. Yeah, so there are many applications. There are applications in forensics, in archaeology, but because here we talk about paleontology, so I just talk about that line.
for we were talking about before we went on air, like how we very much often think about macro paleontology, specifically, dinosaurs or like ice age mammals. And sometimes when we talk about nature, we talk about kind of compartmentalizing everything. It's like, here's this point of time, and then here's this point in time. And we forget that like everything's kind of connected, right? Even with, the tiny microfossils that you study.
Mm-hmm.
How do you think we can go about helping non-scientists, like the general public, think about that better and appreciate looking at the micro world so that we can understand not just the past but the whole world in general?
Yeah, so something that is very cool nowadays with the technologies that we can do, for example, 3D printing of pollen, right? So I don't know if you can see, this is a fossil pond, so that's why it's like splashed. But we can do these 3D models of these microfossils, and maybe start with exhibitions, or when we have like a macrofossil, we can explain that how we know about the vegetation that was around, how we know about the environment. We have the geology and probably some other macrofossils, but we need the microfossils also to tell us information. So for us to reconstruct an environment, we want to use the different proxies available, right? With more proxies, we probably have a more compelling story. But I think the first approach will be through 3D models or to some animation that they can, kids and general public can relate. Yeah, I don't know if we can do like a gigantic 3D impression of a pollen grain to put on a museum. But
I definitely think I would love to 3D print some pollen for myself and just have those. I think those are really cool. Like I said, I looked at, I know a little bit about like foraminifera, which are tiny little oceanic micro organisms, and they find them in the fossil record. And I've seen really cool like art exhibits almost with the 3D printed forms, because the shapes and structures that you see on them are so cool. And from your photos, it looks like pollen and like the little micro fungi have these amazing shapes, and like it's just beautiful art in nature.
Yeah, I remember when I was in Illinois doing my PhD, I was volunteering in a museum. It was the Orphan Children's Museum, and I always give like talks or activities about microfossils to 6 to 10 years old kids. And it was amazing. Like, okay, I put them to play with Play-Doh to make the shapes. I make them to try to understand, for example, how from the plant produce the pollen, or from where that pollen came from, things like that. So to kind of start building that, what is a pollen? What is that I cannot see with my bare eye?
That sounds really fun, actually. I think if you don't mind, I might borrow that for some of our students at the museum.
Oh, just tell me and I send you material. I have material for that.
Awesome.
Yeah. And in fact, in one of the presentations that have all the photos, those pollen grains that were in colors, those were clay made.
Oh, that's cool.
That's awesome. Alrighty. Well, here. Oh, go ahead.
Oh, go ahead. Go ahead, Britt.
Oh, no, go for it.
Well, I was gonna say, like, for because you have your PhD in plant biology, moving forward with like your research as you study like the fungi and pollen fossils, is there like a question that you're hoping to work on more, or like a specific period of time or an area that you want to look more into as a PhD in biology and paleontologists?
Well, so I've been working all over the place. I will always interested in learn more about the Neotropics, the evolution of the plants there. And after one of the projects, the project about the marine incursions in South America, there was this interesting trend that we were seeing, that is, you see a shift in vegetation in South America, in Colombia. So you start seeing mountain vegetation, you start seeing an increase of grasses that has been observed in other places. But this is related with the Andean uplift. So it's to understand more this gap between the Cenozoic until the Miocene and before the Quaternary, so before the last uh, 10,000 years, because there is a gap. And in palynology, we have something that when we are talking about deep time, the type of names that we use for the fossils are different than in quaternary would we use. We will use the modern species name. In the fossil, we don't. We use names like this one, Stratopolis catatumbus. If you don't study or have not reviewed other types of pollen, you don't know which plant or what can tell us, right? So, but there is this period of time, the Pliocene, so that is after the Miocene, after 5 million years ago until 2-3 million years, where we have a shift in vegetation in Colombia. And that's something that I would like to see, like what is happening in the environment there. Like, yeah, we know they change, but how? So being able to integrate not pollen, but fungi and maybe other proxies to have a better idea.
How cool.
That's awesome. All right, one question we always ask any of our presenters is how do you have any advice for future palynologists or micropaleontologists or anybody who might be interested in learning more about this particular field?
Well, just keep your curiosity. You know, I think that's the main thing. Keep the curiosity. If you have a question, no matter how you think that question is, there are no dumb questions. There are not, especially in science. Like, why this plant is growing this way? You know, you can think, oh, this is a dumb question. There is not. Just go and explore, and if you like to go to understand nature, just go out and collect things and look maybe with a lens, or in the school, tell the teacher, hey, can I see this in the microscope? And that's how you build your curiosity. I think that was myself when I was a kid. Being outside, getting with a bunch of bugs and stuff. And my mom like, that's the refrigerator. So just keep your mind open.
Awesome. That's great advice. Brittany and I will definitely agree with that, as Fossil Friday Chats now is just a way for us to satisfy our own curiosity and learn about things.
Yeah. Well, and
for
example, sorry, one more thing, like for the micro world, just take a drop of water and put it in a microscope. You're going to see a bunch of things. So.
Cool.
I think I need to borrow a microscope from my museum. I think I might be doing that. Well, I guess that does it for questions today. Thank you so much, Ingrid, for sharing your story of the microscopic world. I'm definitely going to be reading about microfungi later today, because Brittany, while you were doing your presentation, Brittany and I were texting each other about aren't fungi so cool?
So cool.
So we're definitely going to be reading about that later. If people, you sent us that link, you showed the link for that really cool book, so we'll put that in the description. But if anybody wants to learn more about your work or your institution, where's the best place for them to go?
Yes, so I have my contact information in presentation, but through Twitter. So the Twitter is at Ingrid down dash, I don't know how to say, pollen, or through Gmail, you can contact me. So right now I'm in Morehead State University, but you can email me to my Gmail, that is ingridromero@gmail.com. So, yeah.
Great. Yeah, we'll put all that in the description below, so that if anyone watching, has interest in learning about palynology, you can connect with Dr. Ingrid Romero and learn more about it. Hopefully some future palynologists will be contacting you soon, and we can get more people fascinated with the microscopic world.
That will be amazing. We need more.
Oh, definitely. Every time we have these episodes, I always consider a career change, for a couple hours after. And I was like, I could do it. I could totally do it.
Yeah.
All right. Well, that does it for today's episode. Thank you so much, Dr. Romero, for joining us today. We really appreciate it. And thank you so much to everybody for tuning in. As always, if you like this program and want to support programs like it at the Alf Museum and Western Science Center, you can find links on how to do that in the description below. And make sure you like and subscribe to our channel for more stories from the world of paleontology. Thank you all so much, and we'll see you next week. Bye, everybody.
Bye. Thank you.